Hello, my name is Tatiana. I'm principal scientist in in-vitro neuroscience here at Signature Discovery, and I invite you to join me over these next coming weeks as I will dive how to measure mitochondrial function. We explore the benefits of measuring it, and I will share exclusive data with you as well. In this video today, I will show two different approaches to measure mitochondrial function. One of them is measuring in in-vitro models key and specific energy metabolites related to keep the cells with energy, such as ATP, ADP, NADH, pyruvate, and lactate. I will also show you some internal data regarding oxygen consumption rate, since mitochondria, in order to produce this energy, needs to breathe. So because mitochondria primary function is generate energy, it's really important to keep mitochondrial health, because then we'll keep the whole cellular activities and cell homeostasis, not only the brain, but in your whole body. We can monitor specific metabolites, and these metabolites are produced because we actually have the mitochondrial respiratory chain working efficiently. This information can be very crucial for understanding diseases mechanisms such as Alzheimer's and ALS, especially in this neurodegeneration field, where mitochondrial dysfunction is a hallmark, and evaluating potential efficacy of new therapeutic compounds targeting cellular metabolism. So actually the level of key energy-related compounds and metabolites can be measured through access of bioenergetic health of cell cultures. Here at Signature, we utilize commercially available and standardized off-the-shelf reagents and kits to measure such changes in different metabolites. By employing these ready available kits, guarantee that we use convenience and a very good and efficacy test in conducting mitochondrial function, thereby facilitating the progress of our own experiments. So I will show you some data and some graphics now about how we can measure ATP, NADH, pyruvate, and lactate. So basically in this first picture, we can see different levels of ATP and ADP measuring a neuroblastoma cell model that was exposed previously with a mitochondrial inhibitor called rotenone in two different concentrations. So rotenone is actually used to inhibit mitochondrial function, and therefore, if it's inhibiting its function, the reasonable is that we expect a decrease in the energy production. So in this graphic, we can see that there is a significant decrease of ATP concentration in the cells. Of course, because of that, we also see changes in the ADP and ATP ratios. In this next graph, we can see the measurement of other two key metabolites, NADH and NAD+. So we can see again that neuroblastoma cell line treated with rotenone, again in two different concentrations, do change the level of both metabolites. And the ratio of NADH and NAD also changes. This is really important because, as I said before, the energy part of the cells is not only ATP, but also other molecules that we have inside of our cells. So lastly, I want to show you some data regarding pyruvate and lactate levels. So again, pyruvate is very important because it's one of the metabolites that will enter in the mitochondria to boost ATP production. And lactate will dictate how this conversion of ATP is being efficient. Meaning, like, if you have more production of lactate, this can indicate that something else is going on in the cells. So in the neuroblastoma cells exposed with rotenone, that was the model that we used in these experiments, we can see that actually we don't see quite significant changes despite this increase in pyruvate. However, we do see a significant increase in the lactate levels, meaning that the pyruvate is going to the mitochondria to produce ATP, but still we have a transformation of this pyruvate into lactate, and this can mean that the cells is struggling. So as I was saying before, actually mitochondria to produce ATP, it relies on this what we call cellular respiration. So we have the mitochondria complex chains, a lot of proteins transporting electrons and protons along the mitochondrial membrane, and this is really crucial to maintaining cellular homeostasis. Therefore, if the mitochondria is breathing, if the mitochondria is healthy, we will have an increase in oxygen consumption. if mitochondrial deficiency for some whatever reason, we have a decrease in this respiration. And ultimately, this respiration will have a link with a decrease in energy production anyways. So here at Signature, we measure oxygen consumption rate using two different models. We did use the neuroblastoma cell lines, again exposed to rotenone. This is a mitochondrial inhibitor, but we also use other cell types. Let's go through this first data before. So in the presence of rotenone, this neuroblastoma are actually struggling in keeping their energy level. So we can see in your left hand side, the graphic, where control cells have a higher level of oxygen consumption, while the cells exposed to this toxin actually breathe much less. So if you can see basal respiration, maximum respiration, and spare respiratory capacity, there are 3 different ways to measure oxygen consumption. They are all significantly decreased in relation to the control cell lines. In the next graphic, we measure the oxygen consumption as well, but using MDMI. So these are basically MDMI cells derived from humans. And again, we use the same platform to measure oxygen consumption. In this experiment, cells were exposed to different compounds such as LPS and dexamethasone. And again, we have changes in the respiration, but was not actually significant in relation to the control cell lines, meaning different stimuli can actually induce different changes in different cell types. So this is why another reason is very important when we're talking about mitochondrial function, measure different parameters in your specific model because the data can differ between cell types. If you missed last week's video from our series, visit our website to catch up on how we can measure mitochondrial function and why actually mitochondria is so important for the neuroscience field. And please join me in the coming weeks where I will guide you a little bit more in detail how we can measure mitochondrial function, but using other assays such as kinetic experiments, using live cells. And of course, visit our website to learn more about mitochondrial platform and mitochondrial assays here at Signature Discovery.